We built up an automatic touch force machine to observe the plant thigmomorphogenesis. The robotic arms are installed with human hair brushes to apply gentle touch force on plants. In comparison with human finger touch, the automatic human hair touch machine can offer more labor-saving and uniform touch force loading. The time required for one round of force loading can be restricted within several minutes, as compared to several hours of finger touch and cotton swab touch. The robotic machine can largely improve the plant mechanoresponse research by increasing the efficiency and the population of treated plants. We anticipate that the automatic touch machine will be applied in the force signaling mutant screens and the mechanoresponse studies in both plants and animals.
To begin, test the germination rate of each genotype after nurturing seeds on a 0.8% agar plate with MS medium for 7 days. According to the germination rate, prepare seeds 4 to 5 times more than the desired number of plants to be produced. Immerse seeds in cold water in 1.5 milliliter plastic tubes and cover with aluminum foil to keep in the dark. Store at 4 degrees Celsius for seed imbibition. 5 to 7 days after the imbibition, select the appropriate soil for plant growth. Avoid large clumps and use a fork to mix them homogeneously. Prepare 24 207 milliliter plastic cups with an upper rim.